సౌండ్ వినిపిస్తుంది.
Good afternoon, Principal Madam. Eminent Resource Person, USC Coordinator, Academic Cell Coordinator, faculty members, research scholars, and my dear students. A warm welcome to one-day national webinar on research methodology organized by Department of Zoology, SKR Government Degree College Women, Rajamahendravaram. With immense pleasure to welcome renowned, dynamic personality and great academician, Principal Dr. P. Raghav Kumari Madam. It is my honor to say that she set benchmark in the college history with her visionary leadership. Thank you so much, madam, for giving us this opportunity to organize this type of webinar. Now I request the president of the today's event, Dr. P. Raghav Kumari, madam, to start her presidential remarks.
Thank you, Priyanka. Distinguished speakers, Dr. Joe K. Tizakudan, ARS Principal Scientist and Head of the Regional Center of ICAR CMFRI, Visakhapatnam, and Dr. P. Suresh Babu, Associate Professor, Department of Biochemistry, University of Hyderabad, Telangana State. Participants, faculty, and students, good afternoon and a warm welcome to you all. It is my pleasure to begin this national webinar on research methodology. At the outset, I would like to thank our Honorable Director of Collegiate Education, Dr. Narayan Bharat Gupta, IAS and CCA authorities for permitting us to organize this national webinar. I appreciate the Department of Zoology for taking the initiative to conduct this webinar. I feel privileged to give a brief history of our college on this occasion. Srimati Kandukuri Rajalakshmi Government Degree College, Women Rajamahendravaram, one of the most prominent reputed college leading center of excellence, was established on 2nd September 1968 in the name and memory of Srimati Kandukuri Rajalakshmi, the ideal wife of a great social reformer of South India and a philanthropist, Rao Bahadur Kandukuri Viresilingam Pantul Garu, to empower the girl students with quality of education, ethical values, and culture. The college was under the control of the government of Andhra Pradesh from 10th August 2022 and is affiliated with Adikavi Nannaya University. The college has completed 3 cycles of NAC assessment with a B+ grade and received ROSA funds 2 crores of rupees for infrastructure development. We are committed to the upliftment and overall development of girls residing in and around East Godavari district with a noble vision and mission of spreading the light of knowledge. Our vision is to transform the young women into integrated, empowered, and holistic women leaders. At present, the college offers 10 major undergraduate programs and 1 postgraduate program in various disciplines on its lush green campus. Coming to the today's webinar topic, research methodology plays a crucial role in conducting research studies. It provides a systematic approach to collecting and analyzing data, minimizing personal biases, and ensuring objectivity. A well-chosen methodology ensures that the findings are reliable and valid, thereby maintaining the integrity of the research. A standardized methodology enables the comparison of findings across different studies and populations. A well-structured methodology fosters problem-solving skills, allowing researchers to address complex research questions and overcome challenges. Research methodology develops analytical skills, enabling researchers to critically evaluate data, identify patterns, and draw meaningful conclusions. And also it facilitates collaboration among researchers from diverse disciplines, promoting interdisciplinary research and knowledge sharing. Addressing any research questions often requires the integration of multiple disciplines and the use of innovative research methods. Therefore, research methodology is a way to solve the research problem systematically. The researchers must know not only the research methods or techniques, but also the methodology. Researchers not only need to know how to develop certain indices or tests, how to apply particular research techniques, but they also need to know which of these methods or techniques are relevant and which are not, and what would they mean and indicate and why. Researchers also need to understand the assumptions underlying various techniques, and they need to know the criteria by which they can decide that certain techniques and procedures will apply to certain problems and others will not. The researchers must design the methodology for the problem as the same may differ from problem to problem. In this connection, the Department of Zoology has organized one-day national webinar on research methodology to address the challenges in the research with novel research methodology approaches, to familiarize the research scholars and students with a wide range of relevant approaches to the research, and to provide an in-depth subject knowledge, quantitative and qualitative interdisciplinary research methodologies, thereby enabling the research scholars and students to produce quality work in their respective fields. Before concluding my opening remarks, I would like to convey my appreciation to today's speaker, Dr. Zo K. Kizakudan, Principal Scientist, ICAR, CMFRI Visakhapatnam, and Dr. P. Suresh Babu, Associate Professor, Department of Biochemistry, University of Hyderabad, Telangana State. Hope this webinar will serve as an ideal platform for the students and researchers to interact and explore new methodologies in conducting research and the scope for writing research papers. I once again congratulate the Department of Zoology for organizing such a useful seminar. I wish you all the very best. Thank you, one and all.
Thank you, madam. Now I request Dr. P.S. CHP Deepika Rani, madam, to introduce today's resource person.
Tejasvina Varidamastu. At the outset, my humble pranamas to Dr. Jo K. Kizakudan, sir. Really, it's my honor to introduce the academic journey of Dr. Jo K. Kizakudan, who is an eminent scientist, a visionary, role model of commitment, and highly passionate in research, and he has set a hallmark in the world of research. Dr. Jo K. Kizakudan completed his bachelor degree with first-class distinction at Mahatma Gandhi University, Kerala in 1991, and his master's in mariculture at Cochin University of Science and Technology in the Kerala in 1993, and he did his PhD in reproductive biology, growth, and ecophysiology of mud spiny lobster, Panularis humarus, and the sand lobster, Turnus orientalis, from the Bhavnagar University, Gujarat in 2007. After that, he served as a lecturer in charge and founder, head of the Department of Aquaculture at Sacred Heart University, Tevara, Mahatma Gandhi University, Kerala. During the academic years of 2004 to 2006, he acted as a consultant in freshwater prawn farming for the DST in Kerala and consultant for the shrimp hatchery, Travancore Shrimp Hatchery in Kerala, and for 6 shrimp hatcheries in Andhra Pradesh. After that, he was selected for the National Bank for Agriculture and Rural Development Services and in 1994, and he joined in the Agricultural Research Service in 1996 as a scientist at ICAR CMFRA headquarters, Kochi. And subsequently at the Veraval station, he rendered his services for 7 years, and then at the Chennai regional station for the 20 years, and has head of the Visakhapatnam since June 2023. And now coming to the important areas of his research. He mainly emphasized marine lobster fishery, biology, breeding and hatchery and mariculture, capture fisheries, sea cage farming, environmental impact assessment and rapid impact assessment matrix, halibut biology and maturation and breeding, artificial reef sites, socioeconomic impacts, assessment studies, and reef module development and sea ranching. And the key projects that he used to undertaking are the National Agricultural Biotechnology Project on the dams and ornamentals, and National Agricultural Innovation Project on shellfish value chain and network on climate changes 1 and 2, and All India Network Project on the mariculture, fisheries management for the sustainable livelihoods in its 2 components 1 and 2, and International Fund for the Agricultural Development Post-Tsunami Sustainable Livelihoods Program for Coastal Communities for the Tamil Nadu. And he also acted as DOI for the Department of Fisheries for the NFTV and sea cage demonstrations and artificial reefs. And he is the consultant for the Tamil Nadu and Andhra Pradesh, Kerala, Maharashtra, Gujarat, Goa, and Karnataka, Puducherry, Odisha. And for all these things, he is the DOF. And now coming to the breakouts for the and his silent achievements. He is the team leader for the Dams on Coastal Water Productivity, a document prepared as a prelude to the National River Planning Linking Plan and the potential of marine ornamental fishes at Gujarat coast. And he conducted a number of road rallies and a series of fisher awareness workshops in fishing villages and harbors of Gujarat towards the lobster conservation with the collaboration of the MPDA. And he is instrumental in working the biology of whale sharks prior to the banning of fishing and inducing the administrators and fishers, stakeholders and traders in closure of the fishing practices. And he has also given an interview on the Star Plus TV to Ms. Menaka Gandhi, the Honorable Minister of Environment, on this particular subject. He domesticated and developed many holding systems for the slippery lobsters and slimy lobsters in the country. He also demonstrated the breeding and seed production of the sand lobster, Thennis unimaculators, and hunchback lobster for the first time in India. He developed breeding and maturation systems and a number of techniques for all the Indian marine spiny lobster. Successfully, he demonstrated the open sea cage forming of spiny lobster and the sea bass in the Chennai with the help of BOB with an overproduction of 3.5 tons production. And he successfully demonstrated 20 sea cage units with fisher youth participation forming of lobsters, sea bass in Chennai and Tamil Nadu, and won the best forming award in Tamil Nadu state and ICAR level. Successfully, he demonstrated Indian Pampano, John Snapper, sea bass, mullets, and grouper formings in the coast cages for the benefit of ST-SC communities of Andhra Pradesh, Odisha, and Tamil Nadu. Successfully demonstrated mussels, oyster formings for the fisher communities in Tamil Nadu under the fisheries management for the sustainable livelihoods. He completed more than 400 days of sailing on board fisher boats and crafts and non-research vessels. And he had MOUs with more than 100 fisheries villages, 4 coastal states, and 10 consultancy projects. He completed giving technical guidance for the deploying of over 1,000 artificial reef sites in the coastal states of India, traveled to over 800 fishery villages and interacted with nearly 10,000 fisheries in area. Successfully domesticated the breed in the Indian halibut for the first time in the world. Conducted 2 TOT for the benefit of senior state level officials of DOS and research officers of FSI, SIFNet on artificial reefs for sustainable fisheries. He has successfully released course manuals on artificial reefs for sustainable fisheries in 5 languages. And now coming to the awards and recognitions, he published over 140 papers, 5 course manuals, 20 pamphlets, 1 poster, and 3 research books, and 25 book chapters. He was recognized as a PhD guide in the Madras University Marine Biology and Andhra University Department also. He was permitted to guide a JC Boost Ford Foundation Fellow from College University, USA for the PhD. Guided a high school team in the US for the scientific innovation and application in the environmental conversation and won the championship. And he has visited the Netherlands, Mexico conferences and Australian conferences and Taiwan in connection with the research methodologies, and he has set a hallmark in that particular lobster farming systems. Thank you, sir. Thank you. Really, it's my honor to introduce such a great academician and a scientist, and we are very enthusiasm to hear your talk, sir. Thank you. Now I request Dr. Joe K. Kizakudan, sir, to give his talk. Sir, sorry for the interruption. Sir, we are unable to hear your voice, sir. Excuse me, sir. Sir, we are unable to hear your voice.
Hello. Hello.
Yes, sir. We are audible now.
Yeah, I'm audible now.
Yes, yes, sir.
Okay. Now, are you able to see my PowerPoint?
Now in the screen you are visualizing, sir. PowerPoint.
PowerPoint slides have come.
Yes, you shared your slides, sir.
It has come.
Yeah.
Thank you. Sorry for that. I am traveling. Actually, I am in Chennai now. Since Deepika has been insisting, I am delivering my talk. It is in a hurry. I've just finished my students' Viva in ZSI and I just came back. Good afternoon to one and all. Thank you, Deepika, Dr. Deepika, and the principal and an illustrious college to have invited me. I am new to this place, so sorry, I am not very familiar about the Adikavi Nannaya University much, but I am learning about it. I'm in just 16 months in Andhra. I visited your places several times, and we have a lot of activities there. And good afternoon to all the participants, the faculties, and the students. Research methodology. I want to confess that I was doing teaching for some time, as you heard in the curriculum, and I was also a banker with NABARD for some time. But then my passion was to do more of research and science, so I left all that and I joined the Central Marine Fisheries Research Institute. And my dad was a scientist. My wife is also with me in this division. She's also a scientist. So family, a few of us are into science. Passion runs deep into marine science. So I was into zoology, and then after my UG, I have taken masters in mariculture, did PhD on a marine species, and then further developed it. So research is nothing but what has been there. Either you study more new facts about it on a branch of knowledge. And research is a systematic effort to gain new knowledge, and science is not just belief, but the will to find out. It is a search. It can be research, it can be new search, it can be an old search. It has got two components basically, a basic and applied one. So the basics are more related to theory principles, which is of more open access and has or does need an institutional strength. The fundamental researches take place at bigger establishments or bigger institutes, whereas more of applied and practical solution oriented researches take place at a smaller labs, laboratories, workshops, or even university labs and minds of youngsters. So practical solutions to existing problems, which leads to evaluation, R&D, and action research. This is the way I have streamlined the presentation. So the crux lies in where you are, why you have come this far, why you have done graduation and post-graduation, and what sciences is your interest, and in your research or in your area, what is that left over, what needs to be done, what is the area where your passion lies. If you take physics or maths or chemistry or biology, it's an ocean, and you might be interested or you might belong to a particular area which has got some concern or an issue. So identify the issue, concept, and develop a hypothesis for the various types of research. It can be quantitative, it can be qualitative, just a superficial idea. It could be descriptive, analytical, fundamental, or applied, exploratory, conclusive, and it could be just a survey statistic and analysis of the data, or a case study just to give a reference randomly to say this could be it. So what is research is defining and redefining problems, formulating a hypothesis and objectives, collecting, organizing, and evaluating data, making deductions and reaching conclusions and testing the conclusions. So I have shown a picture which has been drawn by a small child, and this is exactly what I am doing. A small piece of reef I am building on the ocean floor, but it is going away and making huge life in terms of plankton, zooplankton, fish, and so on. The area which I am focusing is a very small area, but the life it is giving is to millions of organisms. And similarly, we have done for thousands of sites. So idea could be small, but it could germinate into a big tree and can make mountains. So science can actually move mountains, and that could solve many of the issues of mankind. And mankind, when I say this, involves a lot of people, a lot of interactions, You cannot be an island and you can be more a fundamental researcher, but then ultimately when it comes to applied and practical researches, the connectivity is also essential. Why to do research? Is desire to get a research degree probably for qualifying people, desire to face challenges in solving unsolved problems, the called intellect people, desire to get intellectual joy in doing something creative, desire to be of service to society, desire to get respectability, directives of the government when you are employed to work and then get a research data out of it. How to do? You have to follow different methods, experiments, treatments, trials, and what are the observations you make. All these are illustrations given just to keep you alive. Post-lunch session would be really drowsy. So you can see laboratory where the guide supervises and how works are being carried out for, say, sediment analysis or water or plankton and so on. And then you come to a subject. Again, I told you your interest is important, your idea is important, based on your past experience or your guide's experience, on your reading and originality. This is very essential. And important features of a research design: a plan, a strategy, and the time and budgets. So you can't just dream of having a big cup of thing in your small budget. So all these are important parameters, and the process are involved into it. How do you develop a project, or how do you visualize a project? Where do you get the funds? Whom to consult? Where to go? And then afterwards, how do you validate your intuitions? You need some senior people also to validate. Your intuitions may turn out to be very silly. Sometimes it may be very crazy, but then you need a crazy idea also to be supported by a senior head. So improve methods. All methods need not be stereotyped. There could be methods where you can improve. That is where the intellectual and creative skill comes in. Demands of the job. Under the job pressures or the university or the institution, there would be a pressure to work on certain areas. Focus will slightly be different. And then subsequently into publication and patents. So, what are the characteristics? As I mentioned, the systematic way of doing it, a logical approach that there should be a principal thesis to be projected and testing of the thesis, and empirical derivations. You need to be sound in your statistics, in your inferences, concordant observations, and objectivity. Why do you want? Whether we replicate and get the same result. That is very important for the upkeep and maintenance of faith on the society, that your experiments, whatever produce results, should be repeatable. It cannot be just one time go and then a guess and a paper is published, which will not be no use to the society. And it should be transmittable, transfer of technology, or at least convey to a next stage where it can convert into a policy or a document where the society or country can benefit. And it can also be a standard for quality controls. So all well designed and conducted research has a potential application, probably for applied research. So define your objectives, try to keep them simple. More variables becomes more difficult. Use the opportunity and get the help at this stage with senior colleagues and experienced teachers. Literature search is something which is very important. If your ideas are really good and concept is really good and you are a crazy idea, probably you will not have much literature on it. You should not be worried. You should be worried when you're taking a track which has been taken by thousands of workers. Probably you will have to read more, understand more, and to spend your time more on literature. So literature is essential, but it depends on what area you're going to work with. A new road you're going to open, probably literature will be the least. But you should all be cautious that the road should not be to make a bomb or something which is negative for the society. Science can be negative at times, but need not be always. But whatever failures you have, if you open up a new road, probably you will be the first one to write an article on it. You will be first, not having many citations, but you'll be referred. So this is the way you have to go ahead. You are not able to see my slides? Hello?
Yes, sir. We are unable to see your slide.
So am I still in the first slide?
No,
Not even one?
no, sir.
happened? Give me a minute. Now, are you able
No.
to see my slide?
No, sir. Yeah,
No, sir.
You have seen a slide?
yeah. The first slide was displayed, sir.
Yeah. Then we move on to the next one. This one you see?
No, sir. Can you please change the next slide, sir?
I have already changed. This is second slide.
No, sir. We are unable to get the second one. Yeah, now.
So probably this is the signals are a little slow. So this was the slide I was mentioning about a small reef area which is growing into a magnitude of organisms, an art done by a small child. So these slides I have already discussed to you how we should develop an idea, how you incubate a concept through public participation, through requirements, or purely through a concept from your mind. This already I have discussed about how do we go out, why do we need to research, and what are the different methods. In this I have discussed how do you choose a subject and the important features of a research design, a plan, a strategy, and the time and the budgets. Till this slide, you are able to see me. Hello? These
Sir?
slides are visible to you?
No, sir. We are able to see only the first slide.
अरे बाप रे, क्या हो रहा है?
Yeah, now we got the second slide, sir.
It is that slow.
Yes, we are able to see, sir.
This is the second slide. Now,
Now it's only the second slide, sir.
there's something wrong. You are able to see the slides moving.
Sir,
Am I still in the second slide or third slide?
nothing was displayed on the screen, sir.
I'm very sorry. This is not working. I think the net is not responding, I think. Hello. Can you see the slides now?
Yes, sir.
Sir, we are seeing only first slide. We are seeing it. Of course, it is ready for scrolling, I think.
Now, second slide, I have already scrolled. That's why I'm asking.
No,
No,
no.
sir. We are able to see
No.
only the first slide, sir.
First slide only we are seeing. Sir.
Second slide.
No.
No, not at all, sir.
I think your Zoom is not functioning well here. Is there any other link you have sent? Only this link, huh?
Just now we got the second slide, sir.
That means it is coming slow. Yeah, what do I do? It is coming now. Okay, let me see that.
Sir, continue your talk, sir.
Okay. Hello, can you hear me?
Yes, sir. Now it's audible.
Can you hear?
Yes, sir.
I think the PPT is creating a problem. Shall I continue?
Yes, sir. You can continue your talk.
Okay. So I proceed to the next slide. I don't know. I'm very sorry. Probably when the mails get connected, I'll send it across to Deepika. We have to define our objectives. As I mentioned, it has to be very simple. Variables can be more difficult, but use the opportunity and get the seniors or your teachers to guide you. That is more important. As I mentioned, literature survey is very important for people who are doing things which are already done hundreds of times. So there needs to be a lot of citation, a lot of references. But if you use a path which is completely novel and nobody has worked on it, it could be just your reference for the first time. So there is always a novelty, but then there is always a risk that you are attempting a program without much of base work. So steps involved: choose a subject, literature survey, define and formulate specific objectives, prepare a synopsis, procure a suitable apparatus or a material or a live animal, design a experimental setup which should be statistically approved or a random design. Then you should be there with the preliminary experiments, execution of the project, accurate measurements and data collection that again should follow some international or national methods and standards. Plus, it should also be ethically correct that we should not build up data for the sake of just completing our thesis. Then sit up for analysis and error compounding, hypothesis testing and verification. What data you observed is true to what you thought earlier. Write your results and discussions with the previous observations, previous workers. Generalize the topic, interpret and draw conclusions in a very small, simple language. Prepare a project or a thesis or a report or a paper. It involves question or a problem development, formulation of goals and objectives, plan design, generation of research outputs, and interpretation of results and drawing conclusions. Research article writing requires very nice language, suitable specified journals, which would really carry forward your message or your research paper. Get a copy of the instructions to the authors, prepare article as per their instructions, select suitable titles, authors and affiliations and abstract, create highlights, novelty statements, originality, create a good introduction such that the readers introduced to the subject easily with the earlier works. The materials and methods, if there is any note, that should be highlighted. Results and discussion should be clear and deliver the message, as well as it should lead to a conclusion even for an ordinary man when he goes through the paper. Finally, acknowledge the source of fund or your team leader or the facility or the infrastructure and the important references which led to the work. And finalizing a research report will include introduction and statement of problem, review of the relevant literature, statement of hypothesis, theoretical resume, description of the research design, description of the experimental design, description of in the entire study. Others need not be brought in just for the sake of giving an authorship. Article is free from plagiarism. It should not be a duplicate, it should not be a copy, and it should follow ethics. And why authorship matters? Because it is a responsibility and accountability of the published work and its implications, which goes down to the society tomorrow. So authorship confers credit and has important academic and social and financial implications. Key principles to writing: you should contribute, it should be a substantial and verifiable data, it should be accountable, it should be responsible to somebody in the society higher in science or a common man, transparent, discussed and agreed upon before the submission, should follow all the ethical standards and do not involve any honorary or ghost authorship such that they don't have any claim. Anything goes wrong, they may say we are not aware. Anything comes as a credit, they may take all the credit. So be careful with the authors, inclusion of author lists. Authorship policies, several criteria are nowadays followed. There are a lot of guidelines given by ICMJ, COP, or credit. Authorship agreements ensure signed declarations before getting into contributions. Despite resolutions, establish an ethic committee to handle authorship conflicts. Plagiarism and ghost authorships should be checked before you get online, international or a foreign or a national journal. Penalties for violation are very severe. Many careers will be lost if your institutions will have debarring from certain journals if you are not clear with it. And therefore, a necessary awareness is also important. Corresponding author is one who handles the manuscript and writes the manuscript, who could also be the project leader or overall communication with the editors. First author can be the primary contributor who conducts the experiment and data or writes the manuscript. Co-authors can be supportive contributors who help in either paper or biology or genetics or data collection or analysis. Last author will be the senior most author. Please note, last author is the senior most author, the project leader, the head or the director or the group leader or principal or your guide. It is not first author which takes the lead. The lead person or the senior most respectable person should be the last person in the list. So this is how you conduct. I had made so many slides on the websites and linkages for connectivity to proper publications and ethics, but somehow my slides, I think you are not able to visit. So unethical practices of ghost authorship, guest and gift ownership authorship, etc. should be avoided. Ethical authorship should be promoted. And very important, again, I come back to the concept, how you make in research methodology. I would just give you a small example and conclude, because if the slides are not moving, I find it very difficult to communicate with the crowd. For students, I would say I was a zoology student way back in 89-91 in Cochin in a college. And I was studying fisheries and economic zoology. And we used to visit our fishing harbors in Cochin and Thopumbadi and other areas. We used to find several resources which were discarded then because there was no export trade for it. And subsequently, my passion was science, I told you earlier, and I started working on getting into the research system, particularly in ocean sciences, and I got into my MSc in mariculture. I completed that and wrote the agriculture research services for marine biology and fisheries. Got selected as a scientist and was assigned the task of working on marine lobsters. Precisely in 89-91, I was working on a discarded lobster called sand lobster Thinus. That time there was no exports and it used to be thrown from the trawling boats. No value in our domestic markets and nobody ate it. It looked like a bug. And this resource we used to take home, do biology, and then understand it during my UG days. And fortunately, in my master's, that was a subject given to me for further taxonomy and studies. And then in my ARS, when I joined as a scientist, I had the opportunity to hold them live, breed them, multiply them, and could produce the technology for hatchery and farm for the first time in world on 2 species of sand lobsters. So this is the passion which has come from a UG, from a PG, and then subsequently my institute also asked for it because these resources were lesser studied and it had got an immense trade potential in other countries. Now we are standing in a position where this is being completely fished out. We have barely very few quantities in the entire coastal India. So I developed a technology. Now a system has been understood. How many larval stages? How do we mate them? How do we hold them? All that has been husbandry practices of sand lobster has been developed. So you need a long concept which has to incubate inside you, then follow it up with the methods. So as I mentioned, there was no road in the case of a new species and in the case of a new technology available for me. Whatever I wrote was the first paper. So obviously my citations will be very less, but I believe that I have done enough justice to the species which has been exploited, which is going down the barrel. So this is how you conceptualize, depending on the atmosphere where you are, what kind of resources you are given, what is your passion and what is your skill levels. If you are a good computer scientist and you have good knowledge in zoology, work on such projects which can be on computational biology or genetics or can be informatics. Or you are good in biology and breeding and handling animals, you should work on breeding and seed production or maturation or understanding the reproductive biology or histology and endocrinology. So it all depends on what is your passion, what is your subject. Read well before you enter into an area and try to see that at the end of the tunnel it is used to develop or even to write in addition to what has been written. Even a 1 millimeter line in your textbook, that could be called science. Revisiting any textbook and writing it again is no harm, and that is research. So you could revisit what has been written hundreds of years back, rewrite them again, but when you rewrite, see that it is not just a duplicate or not just a copy textbook. There should be at least one line added to what you have already learned and you have spent time on, and your understanding on that animal or that metabolism or that process. Additional one line should be added. Then only it becomes science. You can always delete sentences by telling that what was found earlier was wrong. That is also research. When you repeat research, there could be failures, and even failures are footsteps on a research ladder. So never be disgruntled, never be disappointed. Research methodologies will always give you a positive or a negative comment to go forward. It is never backwards. So my advice, have a good vision, have a thorough understanding of what you're learning and what you're going to do. That makes you a good researcher. Then you get into a good team. And the sense of a team is they understand well everybody's capacities and they perform. And that's how it comes out as good papers, publications, patents, and copyrights. With these words, I thank all the participants who have been patient, although I could miss out on my presentation. Thank Dr. Deepika and principal and all the organizers. If time permits and if there are opportunities, I could visit your campus and could directly be of use to you on some occasions. Thank you very much. Good luck to all of you. Jai Hind. Hello.
Thank you so much, sir. Now I request Srimati M. Kasma Madam to deliver a vote of thanks.
You are muted, madam. Unmute yourself.
Good afternoon, sir. Really, we are fortunate to have this scientific webinar session, which has provided a novel approach and a comprehensive review of the research methodology. How to choose an area, time, design of our objectives, essential features of research methodology and budget, process involved in it. Thank you, sir. Thank you so much.
Now I request Miss G. Divyasri Madam to introduce the second resource person.
Thank you, madam. Good afternoon to everyone. It is my great privilege to introduce today's speaker, Dr. P. Suresh Babu Garu, Associate Professor in Biochemistry at School of Life Sciences, University of Hyderabad. He did his PhD from Sri Krishnadevaraya University, Anantapur in 2006. He worked as Assistant Professor in IISER Tirupati, UGC Assistant Professor in Sri Krishnadevaraya University, Anantapur, postdoctoral scientist in George Washington University Medical Center, USA, and the University of Texas, MD, Anderson's Cancer Center, USA. He achieved DST, SCRB, Young Scientist Research Award, sanctioned against of 22 lakhs in 2014, Ramanujan Fellowship Award 2015, selected for UGC Faculty Research Position 2014, 6th Foundation Day for Excellent Service in IISER Tirupati Institute 2021, 7th Foundation Day Award for Excellent Service in IISER Tirupati 2022. He is a member of IQAC, Coordinator for Refresher Courses in Life Sciences, UGC, HRDC, University of Hyderabad 2023, PhD Question Paper Setter 2023, PhD Admission Committee Member, Board of Studies Member, Sri Padmalaya Viswavidyalayam, Tirupati. His contribution to research and development was evidenced by his 54 publications in national and international journals. Apart from the publication, his work shows path to next generation students through his contribution in books. Thank you so much, sir, for accepting our invitation. Now I request Dr. P. Suresh Babu Garu start his talk.
Okay, madam, thank you. Let me share my slides. So please wait a minute. I am having problem in sharing the screen.
Okay, sir.
Can you see the screen and slides?
Yes, sir, visible.
Slides are visible?
Yes, sir, yes, sir.
Okay, thank you. Now, can you see?
Yes, sir, visible.
Okay. So, good afternoon. So first, thank you for inviting me for this talk. So so actually I came to know that there are UG students, PG students, and PhD in the conference, that is in the seminar. So that's why I framed the slides based upon how to design an experiment, design hypothesis, design the experimental approach, all these things. Okay. So my today's talk is research methodology for life sciences. So how to understanding the scientific approaches and techniques. Okay. So what is this research methodology? Why we have to use research methodology? Okay. It's a systematic approach to conduct scientific research. Okay. So how we will conduct a scientific research? That's what, it's a systematic approach. It involves first designing a problem, defining a problem, and based on answering that problem, we have to design the experiments. Once we design the experiments, we will get the results, and that result should be analyzed. So research methodology involves conducting a scientific research and defining a problem, designing experiments, and analyzing data. Okay. So what it will give you is that it will help you in identifying new drugs, understanding the molecular basis of the disease, as well as basic research and biotechnology advancements. Okay. Okay. Now, what are the types of research in life sciences? Is that basically we can categorize into 4 types. One is basic research, applied research, clinical research, and translational research. In basic research, what we do is that we will understand the basic concepts, molecular mechanisms. Say, for example, how a cell division is happening, how a cell divides, how the biological functions are happening, what are the signals that are responsible. Like that, we will understand the basic mechanisms. So that is important to develop the knowledge. And example, molecular biology, we know that replication process, transcription process, translational process, all these things we study. And the experiments related to that involves basic research. Then applied research. Applied research is helps in it's in a practical applications, that is it will help you for a discovery of a new drug, new concepts, and it is directly helpful for what is called as human beings or animals or plants. Coming to the clinical research involves human subjects. Say, for example, if I identify a drug, so it can be tested on humans. That is a clinical research. Then translational research. Generally, what we do is that we take chemical compounds, we take plant extracts, or we take some other chemical inhibitors. First, initially, we establish these findings in cell lines. Then we will take it to the mouse models. Then we will take it to the human subjects, isn't it? So this is a translation. The findings that we have in the lab can be can bring it to the medical practice. That is a translational research. Can you hear me? Can you hear me?
Yes, sir.
Yeah, thank you. So in research process, there are several steps. First one is identify a research problem. How we will identify a research problem? Second thing is that literature review. Then third one is formulating a hypothesis. Fourth one is designing the experiment. Fifth, data collection and analysis. Sixth one is conclusion and publishing the findings. So what is literature review? Why it is important? Say, for example, you are, say I am working on cancer biology. So if you want to understand what is cancer biology, you have to read lot of literature. Okay, what is cancer? How many types of cancers? Okay, so far what has been done. So if you review all the literature, it helps you to formulate a logical hypothesis that that is one thing. Second thing is that what are the gaps in the area? What are the research gaps? What has not been discovered so far can be identified. Then another thing is that, say, for example, the idea that I got here, maybe someone has got somewhere else. They might have already done it. So it will prevent the duplication of research. That is very important. So understanding the concept and review of literature is very important. Then where from we will get the information for this is that there are several journals which are publishing the findings. So you will get it from the PubMed or Nature or science journals like that. There are several books, conference proceedings, and there are online databases like Google is there, Scopus is there. Nowadays, we have this ChatGPT. Now we have DeepSeek. Like that, there are several, online sources are there where you will come to know about the, information about a particular thing. Okay. Then formulating a hypothesis. So if you are hypothesizing, say, for example, a gene, an oncogene is there. Okay. What is an oncogene? Oncogene is the one which is responsible for cancer progression. So like that. So if you want to say, you know what is the gene first initially, then only you can say it has a role in cancer, isn't it? For that, first thing is that null hypothesis. Null means you will say that it may not have any effect. It doesn't have any relationship in cancer. Okay. Alternate hypothesis is that expected effect or relationship. That means you will have some preliminary data that maybe it is overexpressed in cancers, it is showing positive correlation with proliferation, or it is showing positive correlation with the genes involved in invasion, migration, EMT. So then you have a preliminary data, then you will know what will be the expected outcome. Then only you can go for alternative hypothesis. Null hypothesis, help you that you are not, it is not misleading. You will have some statistical data which will confirm whether the effect is true or not. So you will you will start with no effect or no relationship. So there is an unbiased way of doing research. So characteristics of a good hypothesis involves the hypothesis should be clear, testable, and measurable. So these are the things that you have to keep in mind. Okay, then research design in life sciences. So first one is experimental research. We know that we have controlled experiment in labs. In labs, what can be done? We have the cell line models, and we have mouse models, and we have in the mouse also we have transgenic models or knockout models, like that, isn't it? So we will test. Say, for example, we will take the signal and we will see what is the impact of the signal in the cells or in cell lines or in the mouse models. So that is a controlled experimental situation. Okay. And you will divide into groups like, what is a control group, what is a, treatment group, what is a, disease model, like that. Next, observational research. Observational research depends upon field work. We will go on field as well as in surveys. Okay. Then we have the clinical trials. So testing drugs on human subjects is a clinical trials. Comparative study. So analyzing, different data sets from online or available resources. So genetic variations, how these genetic variations happen within the species or different species, like that. That is a comparative study. Then data collection methods. How we will collect the data is that we have two types. One is qualitative data. Qualitative data, how we will collect? It is based on the interviews or based on case studies or observations. That is the qualitative data. Quantitative data means when you do the experiments, surveys, and it is having statistical significance, that gives you the quantitative data. So in life sciences, how you will generate the data is that by using different programs like PCR amplifications, gene sequencing, Western blotting, Northern blotting, microscopy, imaging like confocal imaging, immunohistochemistry, spectrophotometric analysis like UV spectrophotometric analysis, chromatographic methods like gas chromatography, gel permeation chromatography, ion exchange chromatography, using cell culture models as well as animal models like that. By using these techniques, we can understand we can generate the data. Okay, so that is only the quantitative data we can generate, not the qualitative data. Okay, then data analysis. How you will be significant about your data is that, so initially what we do experiments, isn't it? Always experiment has to be replicated 3 times, and always we will keep the samples in triplicates, isn't it? Then only we'll get meaningful data. But when you are presenting the data, you have to have statistical significance. How you will derive the statistical significance is that by calculating mean value, median value, and standard deviations. Then say, for example, if it is between 2 groups or 3 groups, what analysis has to be carried out, you should have an idea, statistical idea. So for that, you will use the t-test, ANOVA, regression analysis, like that. So that this will give you the statistical significance. It's not that first time you will get 10-fold change, second time you will get 100-fold change, third time 1000-fold change, means it doesn't have any significance, isn't it? So like that. Nowadays, there are several software tools which are available in the market in order to confirm the statistical significance. Like Python is one model, GraphPad Prism, bioinformatic tools like BLAST, Clustal Omega. These can be used in order to analyze the sequences among different species. Okay, now ethical considerations in research. This is very important. So far, what we have done is that how to design an objectives. That means you have to read the literature, then we have to hypothesis. Then once we have the hypothesis, we will design the objectives. To the objectives, we will have the experimental plan. After that, we will do the experiments in such a way that you will get statistically significant data. Once we have the statistically significant data, we will go for publications. But in order to start the experiments, we need some permissions, ethical clearances. One is IBSC, Institutional Biosafety. Why we require institutional biosafety is that, say, for example, you are using a animal, that is a cell line model. But there, what you are doing, you may be introducing a gene for overexpression purpose. You are deleting a gene for studying the importance of the knockout of that particular gene. That means functional validation. At that time, so you are manipulating the gene at the cell line model. For that, you need to take institutional biosafety, IBSC. Okay, anything. So say, for example, you are working with plant samples, animal samples, anything you want to work in the bench, in the lab, you have to take the institutional biosafety. Okay, especially in order to clone a gene, all these things, you need institutional biosafety. Say, for example, I have the reagents like rDNA, that is cDNA, which has been cloned in overexpression vectors, or what is called as overexpression vectors. I want to send it to some other person who is doing research on that object. At that time, there should be a material transfer agreement, MTA. So all these things you require. This is called as institutional biosafety, IBSC. Okay, then say, for example, you are doing diabetic research or you are doing cancer research. We want to directly do on tissues. At that time, so we may request the patient, can you give a blood sample? At that time, we have to take the person's consent that your blood has been used in order to do all these parameters, whether you are okay with that or not. Say, for example, at the same time, so tissue samples, you are collecting a tissue from a patient. At that time also you require a consent. That means whatever the human sample that we are going to use, we have to take the consent from the person. Okay, then only we can collect the samples from the clinicals. At the same time, say, for example, I am working on cancer samples. Cancer samples, where from I am getting? I am getting from a hospital. So there we have the ethical committee, which we call it as institutional ethical committee. So first thing is that hospital should get institutional ethical committee in order to procure or in order to get the human samples from the patients. And if they are giving me the samples, means I should also get the ethical that is institutional ethical clearance from my institute or university. Then we should have MOA or MOU between the hospital and between the university. Then only that person can transfer the tissue to my university. Otherwise, it's unethical. So that means ethical clearance is very important. You cannot directly collect the sample from the patient and take it to the bench and work in a university or institute. You require institutional ethical clearance. Okay, you got it. Say, for example, you are working with RNA or protein samples of the human patients, sample patients. At that time, you don't require any animal ethical clearance. Only if, say, for example, if the hospital will take the human sample, they isolate the protein, they isolate the RNA, and if they give it, then you don't require anything. MOA is enough. That is a memorandum of agreement is enough. Okay, then animal research ethics. Animal research ethics includes, say, for example, how you are going to handle the animal. So what conditions you should have follow. So there are certain rules, that we have to follow while handling the animals without pain. How you are going to sacrifice the animal. What are the conditions that you are maintaining? Whether, what are all the treatments? How many animals, you require? Okay, so whether they are wild type, whether they are knockout, whether they are transgenic, or they are induced for a particular disease like diabetes or mouse model. Okay, how you are sacrificing it, how you are maintaining it, how you are withdrawing the blood sample, how you are taking the tissues. So all these things you have to mention, and you have to get the ethical clearance. You should not violate any ethical problems. So these are the three important things that you require. One is IBSC. If you want to work with cell lines or plants or tissues or read RDNA technology, that is a plasmids, okay, or microorganisms, you require institutional biosafety. There also there are two stages: BSL-2 facility, BSL-3 facility. We know that BSL-3, even with virology, that is a pathogenic models, if you are working, you require BSL-3. If it is BSL-2, laminar hood or biosafety cabinets you are working, then you require BSL-2 facility. Then what is the that is a data fabrication. So that means once you collect the data, so I just now I mentioned, okay, that experiment should be carried out minimum 3 times. Each sample should have triplicates. Okay, then only you will see, the reproducible. And the most important thing in research is that reproducibility. Then only it will be considered as an experiment. The data will be, you can trust the data. So that means you have to have consistently the same result for 3 times in 3 triplicates. Then only it is trustworthy. Okay. Then plagiarism. Plagiarism means you should not copy any sentence. You should not copy any data. As Dr. Joe was mentioning, this data fabrication and plagiarism is a very, it's very unethical, and it will lead to serious, issues. Then there will be institutional review board for approval. Say, for example, I will put up a project, say that this, I want to work with a particular gene in cancer. I'm going to use, cell line model. I'm going to use animal model. I'm going to use what is called as human samples. Then institutional review board will go through the protocols or procedures and what experiments that I am doing. If they approve the proposal, then only I have to carry out the experiment. Without this, if you do any experiments in the labs, that is unethical. You are punishable. So that's why your proposal should be approved in the ethical board. All these, ethical clearances should be there before proceeding with the experiments. In some institutions, they made it mandatory. Even if you get the, if you want to buy an animal, mouse model or any other model, you should have animal ethical clearance. If you want to buy any cell line culture, cell lines from ATCC or MTCC or from, there are several sources, you need IBSC. Okay, if you are asking for a human sample, you need institute ethical clearance. So they made it very strict, and it is mandatory. And even tomorrow, if you are publishing a paper, you have to show all these clearances. You are publishing, you are going for submission of a thesis, then also you have to show this. Okay, then writing and publishing. So how to structure a research paper? Now what you have done is that first you have reviewed the literature, identified the research problem, there is gaps, then hypothesis you made, then based on that you have objectives. In order to fulfill the objectives, you have carried out the experiments which has statistical significance. Then after that, you actually conducted the experiments, then you got the results, analyzed, okay, and analyzed data you need to publish now. Okay, how significant it is, you all statistics will tell you. Then if you want to publish a research paper, what needs to be there is that first thing is that you have to give appropriate title for the manuscript. Then authors, every author should have significant contribution in the paper. As Dr. Joe has mentioned earlier, it is not that never ever give a free ride. He may be a friend, he may be your a brother or sister, never ever give a free ride in research. Okay, then what the abstract will give you in a research paper is that abstract will give fine findings, what you have achieved during your research. That is the abstract. Then how to write an introduction? How to write an introduction means introduction should be given to your results. Say, for example, you have achieved, you have identified a gene which has a role in a cancer progression. Then what you have is that you have established, cancer, it is progressing through apoptosis, that is a proliferation assay or MTT assay, like that, isn't it? So your result, that is based on the results only, you have to give the introduction. It's not that you can give very vaguely. No, introduction should be given to the work that you are doing and to the results that you have achieved, so that they will get a reader will get a clear idea why you have done, what results you have achieved. Then materials and methods, clearly specify what are all the methods that you have used. So along with the catalog number, company name. Why it is important is that, okay, you have to use the analytical grade. Tomorrow if I use some other chemical, I may not get that results. So that's why always, in order to ensure the reproducibility, it is very important to note down the material along with its catalog number and the source. Then methods. Method should be reproducible. So the method you are using should be, you have to give complete details about the methodology. You may be following someone's methodology, but you have modified according to your work nature, isn't it? So it's not that, oh, I have carried out the experiment as suggested by so and so, okay, as described earlier. Not like that. You might have changed, isn't it? So accordingly, your methodology should be there. And whomever in the world, if they do or repeat the experiments under the same conditions, you must be in a position to, they must be in a position to do it. That is very important. Otherwise, say, for example, if you publish a paper saying, given a method, if someone in the world, they don't get it, always they can approach you saying that we have used the method that you have prescribed or described in the paper. Can you give explanation why I am not getting? So that's why it is very important to mention the concentration of each and every buffer that you have used, what conditions has been implemented. All these things are important. Then the results. So now you have carried out the experiment. Now you will get the results. So each result, you just briefly what you have achieved, what the experiments is telling about is a result, isn't it? Then you have to write the discussion. So how you will write the discussion is that you have to discuss the results that you have. Earlier, anybody got the similar type of results, okay, in the same model or in a different model, then what it will tell you, what importance it has. So all these things we will discuss in the discussion section. Okay, then conclusion. So how we have to conclude, what is the importance of that, your research in maybe 2-3 lines. So maybe the words that you have used in abstract or the sentences used in abstract or introduction and result and discussion should not be the same. it should tell you differently. Okay. Then references, you have to quote whenever you give a statement, it should be given reference. Whenever you are using a particular method from some paper, you have to, it should be cited. Okay, methodology and I think there is some sort of choosing the right journal. So here thing is that if you are doing a research in diabetes, so you should be published in a diabetic related journal. If it is cancer research, cancer published journals only you have to choose. We just especially people love to publish in society journals, like if it is a basic research like JBC, BBRC or FEBS letters like that. Choosing right journal is very important. Why? Because it will give you your research will go to different people who are working in the same area. Say for example, if you do work with animals and publish in a plant related journal, no one will look into your journals, isn't it? Then it is very important to always have or select a peer reviewed journal, so that it will be reviewed by 2-3 experts, and you will get the what is called as comments. And if you answer those comments, it will be the paper will become very strong. Then what are the challenges in life science research today is that first thing is that funding. We are not getting appropriate funding from either for different funding sources. What are the funding sources nowadays? We have DBT is one, SERB is one, ICMR. Okay, like that, these are the major funding sources in India now. So we are not getting enough funding. Why? Because funding has become very difficult now because of the competition. Next, reproducibility crisis. This is the most important thing that we need to address. What we do, it is not that we do experiments and we publish it, but if someone do that, it should show the same result, but somehow it is not happening. Okay, next, interdisciplinary collaborations are very important. So you may be very good in research, but say for example, I am a molecular biologist. If I want to do biochemical research, definitely I have to collaborate with a good biochemist. If I want to do a microbiologist, I have to go with a microbiologist. If I want to go for RNA-seq or next generation sequencing, I need a good bioinformatician. So like that, now it has become interdisciplinary. Collaborations are must. Okay, it's not that I cannot do whole and soul everything. Okay. Next, what are the future trends in life sciences? Now, it is very important to understand what is the importance of this artificial intelligence in research, how it can be used, what purposes, whether it is for research planning or for hypothesis, for framing the objectives, or for experimentation. There is one thing. Then CRISPR and gene editing. CRISPR and gene editing. So for generation of the knockouts or for generation of knock-ins or transgenics, we need to have CRISPR, gene editing, what is called as techniques in place. Then personalized medicine and genomics. Nowadays, what is happening is that with the advent of genomics, proteomics, and this RNA sequencing, we know what are all the genes that has been responsible for the progression of that particular disease. So once we have identified the upregulated genes and the downregulated genes and the what is called as a fingerprint of that disease, we can target that particular disease by using a particular drug or a specific drug, that is called as a personalized medicine. So in conclusion, what I want to say is that research methodology is the foundation of scientific discovery. Rigorous research tools leads to credible and impactful results. Ethical consideration and innovation are the key to future advancements. Thank you. If you have any questions, I am open to take the questions.
Thank you so much, sir. Now I request Dr. AKP Shivaranjani Madam to deliver vote of thanks to Dr. Suresh Babu, sir.
Now, if you have any questions from student side, I will be happy to answer the questions.
Now the session is open for questionnaire.
From UG students or PhD or postdocs? Okay, then, thank you.
Now, I take immense pleasure to extending my sincere thanks to Dr. Suresh Babu sir for accepting our invitation, even in his busy schedule. He highlighted the key issues, recent developments in research methodology, started with the basics and explained the modern technologies, including the bioinformatics, CRISPR, AI, and ChatGPT. He started with the formation of a hypothesis and ended with the application of your research findings all together in a nutshell. And especially he highlighted the ethical considerations while performing the research. Thank you so much, sir.
Thank you.
Now vote of thanks by Dr. Priyanka Bhatt.
Now it's time to conclude the session. I would like to thank Dr. N.B. Gupta, sir, Director of Collegiate Education, Government of Andhra Pradesh, Dr. C. Krishna, sir, Joint Director of Collegiate Education, and Dr. S. Shobha Rani, Madam, Regional Joint Director of Collegiate Education, for their support. I extended my thanks to our beloved principal, Dr. P. Raghav Kumari, Madam, for her motivation and guidance. With under your guidance, Madam, only we are able to conduct this type of webinar. I extended my thanks to eminent resource persons, Dr. Joy Kizakudan, ARS Principal Scientist and Head of Regional Center, ICAR, CMFRI, Visakhapatnam. Thank you, sir, for your informative session. Enlighten your source of knowledge is amazing. Thank you, sir. I would like to thank Dr. P. Suresh Babu, sir, Department of Biochemistry, Associate Professor, University of Hyderabad. Thank you, sir, for your informative lecture and valuable time. I extended my thanks to Dr. P.S. CHP Deepika Rani, Madam, for her dedication and cooperation. She is the initiator and played key role for the successfully completion of this webinar. Thank you so much, Madam, for your arrangements for this webinar. I would like to thank Ms. G. Divyasri, Madam, for her continuous support and teamwork and responsible towards the department. I extend my thanks to organizing members, Srimati M. Kasma, Madam, and Dr. AKP Shivaranjani, Madam, for their support and affection. I would like to thank technical support, P. Subramaniam Garu, Maunika Mahat, Madam, and Bhuvaneshwari, Madam, Department of Computer Science. Thank you so much. Now the session is ended.